Good evening everyone. This is your 11th week session. So in today's session we will be talking about the topics related to week 11, in which you have looked into the protein engineering, in which you have looked into what is protein engineering, how we do protein engineering, and which are the things or aspects of protein which can be engineered, and then how to make a logical protein engineering, in which you have looked into different methods of protein design such as rational design and different stories behind it. Along with this, you have focused into de novo design of proteins and other directed evolution based methods and different challenges which are associated with these methods, and what are the future directions in the field of protein engineering. Then we will practice few MCQs and we'll also look into the literature, and then I'll take your doubts. So let's start with the session. So if we talk about the proteins, then these are the macromolecules which are made up of one or more long chains of amino acid residues, as you can see. So these residues, this is a long chain which interacts with one another and get folded into a particular structure. So this is the energy landscape, and the least energy structure represents the one which is in the native state or which is most stable structure. So the ultimate aim of a protein is to get into this global minimum structure, which is the most stable structure. So each protein sequence of amino acid is determined by the nucleotide sequence of its genes. So from the 3-letter code from the nucleotide, it determines that what would be the amino acid residue, and ultimately different amino acids decides that what will be the protein fold. So this sequence, it drives the protein folding into a specific 3-dimensional structure which will ultimately determine the activity of that particular protein. So now what is protein engineering? So it involves improving the function of existing proteins, or we can also create new functions depending on the need by altering their amino acid sequence. So we can alter the amino acid sequence, we can make certain mutations or changes in the protein so that we can enhance its function. So this discipline focuses on tailoring the proteins for specific tasks or enhanced performance. And then the technology which can be used for this protein engineering, it is recombinant DNA technology, which is key to this protein engineering. So what we do in recombinant DNA technology, we allow specific changes in the amino acid sequence and we increase its production. So for example, if we have an ordinary protein, then with the help of protein engineering, we can make it a switchable protein. That means depending on the input, it will have some effect. It will undergo some change and produce a suitable output, which could be in the terms of biological activity. It could either be gene editing, or it could act in the terms of biosensing or cell signaling, transcription, pathogen detection. So these are different inputs. With respect to them, certain output is produced. So this type of engineering in which we decide that what our switch will do is known as protein engineering. And to engineer such proteins, we perform the recombinant DNA technology, in which what we do is we have the DNA sequence, we make some mutations or some changes which can further bring about the change at the level of proteins. And now if protein structure is changed, then its activity will also get changed. So that is the recombinant DNA technology, and in the later slides we'll see that how it is done. And now what are the applications of this protein engineering? So it is mostly used in biotechnology to design certain drugs or proteins, or to increase the production of certain products, such as we can increase the productivity of enzymes, which can be used for different purposes. Then we can also use this engineering in medicine and agriculture also. So we can design certain plants, or we can enhance the traits of certain plants so that they can deal with extreme conditions, such as they can be grown in the conditions where there is water scarcity or high temperature conditions. So according to the recombinant DNA technology, we can make those changes in the protein, or make those changes in an organism such that it can give better activity. So this all comes under protein engineering, in which we are engineering the protein. So now if we talk about the protein architecture, then there are 4 different levels. First one is the primary structure, in which we have a linear sequence of residues which are present and they have certain interactions between themselves. Then comes secondary structure, when this primary structure, it makes hydrogen bonds and adopt a structure. So this is a small structure. This could be alpha helices, beta sheets. So this entire protein, it can have multiple alpha helices or beta sheets depending on what are the residues which are present and how they are interacting with one another. and then the secondary structure can further get folded into a tertiary structure where it get folded. So it involves further folding, and different other interactions come into picture, such as hydrophobic interactions, disulfide bonds, ionic bonds. So along with hydrogen bonds, all other interactions also come into picture in tertiary protein structure, which is a three-dimensional structure of a folded protein. And then comes quaternary structure, in which if a protein have multiple units or multiple monomers, dimers or trimers, so those multimeric units, they can combine together and then they make a bigger structure. So this comes under quaternary protein structure. Now along with this, a protein can also have super secondary structures. So this comes after the secondary structure. For example, in our protein, there could be a combination of multiple secondary structures which are observed, such as helix turn helix. So this is a combination of a secondary structure which is quite unique for a protein. So this comes under super secondary structure, as it comprises of a localized motifs of secondary structures. So motif means these are the repetitive units. So in an entire protein, you can find these super secondary structure occurring multiple times. And then these motifs, it gives the protein unique structural features which enables their unique functions. So these specific super secondary structures, they can, although they vary in their structure, so they can also bring about new functionality to a protein. So by identifying such super secondary structures, we can determine that what is our protein and what kind of activities it can perform. So multiple types of super secondary structures are alpha helix motifs, in which it involves alpha helices, such as helix turn helix. Then another one is helix loop helix, and then coiled coil. So these are some types of alpha helix motifs. Then there are beta motifs, in which multiple beta sheets are present. So they could be beta hairpin motif, Greek key motif, which appears like a Greek key, and then beta barrel. So this is also, it also involves multiple beta sheets which are present next to each other and forming a structure like a barrel. Then there are combination of both the alpha helices and beta sheets, which give rise to alpha plus beta motifs, such as beta alpha beta or zinc finger motif. So these are some of the super secondary structure. And why we are going into protein architecture? Because if we want to engineer a protein, then we must know the basics of a protein, that what are the different, how the protein is getting folded and what are the different factors responsible for it, so that if we know the background information about the protein, then we can make certain changes as per our need. Now the second thing is the torsion angles in the proteins. So torsion angles, they are also known as dihedral angles, and these are the angles which are present between two planes. So they are made between four different atoms and two planes. So these four atoms, they are sequentially bonded to one another in proteins, and they describe the rotation around the bonds in the backbone and side chains. So they determine that what will be the three-dimensional structure of the protein. And if we know these torsion angles, then we can determine that what will be the three-dimensional structure of a protein and whether those angles, whether they are feasible or not. So they also determine that whether our protein will be functional or whether it will be stable. So there are multiple such torsion angles, such as phi, psi, omega and chi. So omega angle, it is typically 180 degrees, which means our protein is in trans conformation, and if it is 0 degrees, then that means our protein is in cis conformation. So it simply represents the peptide bond planarity. So for example, let's start from the structure. So if we have these planes, that is plane between C I minus 1, that means the previous carbon which is coming from the amino acid residue at position -1, and then the current residue which contains the nitrogen NH group, and C alpha I, that is C alpha bond of that particular residue which we are considering. So the angle which is formed between these planes, this particular angle is known as phi. So this is phi angle, and then comes psi angle, which is between these planes, this orange one and the pink one. So between these four atoms, the angle which is formed is known as psi angle, which is with this C alpha and then C, and then the C double bond O and the next N. So this type of bond which is between C alpha and C, it is psi, and the bond between N and C alpha, it is phi. And the omega bond comes when we have a peptide bond, so that is C double bond O and NH. So the bond between these two planes, So the plane of C double bond O and NH plane, so that is known as omega because it is determining the peptide bond. And it also governs that what will be the orientation of our protein, whether it is in cis conformation or trans conformation. And these psi-phi angles, they also determine that what will be the structure of the protein. So we'll see in the next slide about Ramachandran plot, where we'll see that how these angles determine the secondary structure of a protein. And this is also just to show you. And then we have also discussed here the chi. So the chi angle, it is between the side chains. For example, we have a C, and then this particular side chain, this can have another means this carbon is attached to other carbons. So the bond between the side chain residues, it is known as chi bond, and which will tell you that what are the side chain torsions. So this is not means majorly this one is not studied. Majorly we focus only on the chi and psi bonds. So phi and psi bonds instead of this chi bond. So if we talk about the Ramachandran map, then it displays the allowed regions for phi and psi angles. So if we have an unfolded protein, and if that protein is of 100 amino acid residue long, then there is a possibility that it could adopt more than 3 raised to power 100 conformations if we consider the secondary structures to be of 3 types, such as alpha helices, beta sheets and turns. Then there is a possibility that each residue can adopt any one of the conformations. So now it's very difficult to decide that what will be the native protein structure. So this is not the case. That means there is these all conformations are not favorable. And then this leads to another paradox, which is Leventhal paradox. That means there are multiple such conformations which are possible, but there are only few conformations which are low energy folds and which are favorable. So instead of exploring the entire landscape when we are studying the protein folding, we will only focus on those energy folds which are the low energy folds or which are the compact structures. So to solve this problem, Ramachandran has introduced a plot in which he has mentioned the phi and psi angles, and the value of each angle will determine that what is the favorable or allowed secondary structure. So most angles or most combination of phi and psi angles, they are sterically disallowed. Sterically disallowed means each amino acid, it has a structure, it has certain atoms which are present, so it takes some structure. So if we have a bigger amino acid, then if there are two bigger amino acids which are lying next to each other, then they will sterically unfavorable, they will repel each other, so they are not sterically allowed. So in that way, psi and phi angles, they determine that whether the residues which are lying next to each other, whether they can take particular secondary structure, whether this fold is or whether this angle is favorable or not. So allowed regions are, for example, in this case we can see the top left. So this particular area which is represented in this particular color, it represents the beta sheets. So if phi psi values lie in this range, then they will give rise to a beta sheet. Whereas in this region also it will give rise to beta sheets only, but these are less favorable region and this is highly favorable. Whereas the white spot which you can see, the white region, so this is the disallowed region. So if you have phi psi values in these regions, so that means that they will not adopt a proper secondary structure, and this means if in your protein these angles are present, then you have to further refine the structure. And then this particular region, which is the bottom left region, it represents that if phi psi values are present in this region, then they will give rise to the right-handed alpha helix, and if they are present in the top right region, then they will give rise to left-handed alpha helices. And this is also the representation of Ramachandran plot with some other means this has divided this entire Ramachandran plot into different regions. So if values of phi psi lie between this range, then they give rise to right-handed helices. If they lie in this range, then they give rise to left-handed alpha helices, and then again right-handed and then left-handed towards this region. Whereas the whitish part, it is highly unfavorable, whereas this bluish part, it is still favorable, and this pinkish part is the one which is highly favorable. So if our psi phi values lie in these regions, then we are quite confident enough to predict the secondary structure. And if we have obtained already a protein structure by X-ray crystallography or NMR, then we can verify the structural stability by looking the value for phi psi in this Ramachandran plot. So now if we talk about the evolution in proteins, that how the protein have evolved. So mainly enzymes, they have shown the evolution throughout time, and they drive adaptations, optimization and innovation in living systems. So if we look around and compare human beings to the primitive organisms starting from the prokaryotes, then we can see that there are multiple adaptations through time which has happened, and mostly in the enzymes or in the proteins which give rise to multiple enzymes. So different machinery have advanced, and the basic basis for that is the evolution, and mainly the proteins which evolved were the enzymes. So evolution acts through diversification and natural selection. So not all the mutations or all the changes which have happened, they are not favorable. Certain changes which are not favorable, they were lost. So there is natural selection process which is acting on this evolutionary process, which affects the complexity from single proteins to ecosystems. So for example, in this case, as you can see, we have these proteins. So this particular black bar in the middle, it represents the threshold, and these are the delta G values. So delta G of folding. So if delta G value is towards the left side, that means if delta G value is negative, then that represents that our protein structure or the enzyme structure, it is stable. Whereas if these values are towards the right, or if these values are positive or towards this threshold, more than the threshold, then it means our protein structure is unstable. So proteins in the parent population, it has a stability of -10 kilocalories per mole. Now there are some destabilizing mutations which took place, and delta G is 3 kilocalories per mole. That means the changes which has happened, so the difference between both the energies is 3 kilocalories per mole. That means it has shifted from -10 to -7. That is this is the difference in the energy values. So now this -7 is still less than the threshold. So this mutation is favorable. So mutation is selectively neutral since mutant protein satisfies the stability threshold. So nature has decided some threshold value, that if our delta G value lies below that threshold, then the mutation will be selected. So natural selection will accept that mutation. But if in the second scenario, if you see, we have all the proteins which are lying in this -7 kilocalorie range. Now if further second round of mutation happens, so then what will happen is, if again its delta G value is 3 kilocalories per mole, then in this case it will cross this threshold and it will be at the value of -4 kilocalories per mole, which is greater than -5. So in this case, whenever the mutation leads the protein passes the threshold, then this mutation is culled by the natural selection, since mutant protein fails to specify or satisfy the stability threshold. So in such scenarios, this mutation will be lost. So natural selection will act on this and purify it. It will remove that particular mutation which is leading to the unstable protein structure. So this is how the proteins have evolved. Protein stability and mutations. So protein stability is measured as delta GF. So this one delta G, this is the Gibbs free energy of folding. So if there is lower delta G value, that means our protein is stable. And if there is higher delta G value, that means protein is unstable. So destabilizing mutations are the ones in which the delta G values are greater than 0. So which means that the energy value is moving towards the higher range. That means from -10, if we are moving towards -9, -7, that means we are increasing the energy. We are moving towards the positive values. So that is known as destabilizing mutation. And if this passed a threshold, then that particular mutation will be unfavorable mutation. So if stability remains above threshold, mutation is selectively neutral. Protein retains the function and stability, and if stability drops below the threshold, that means in this case this is the threshold value. So if it moves towards here, and if threshold value is not met, then in such scenarios this mutation is removed by the nature and protein will become non-functional or unstable. So in such scenarios, unstable or non-functional protein is not kept with time, it is lost throughout evolution. So the mutations under direct selection, so if it is a beneficial mutation, then it will increase and it must be favored to be a beneficial mutation. And if it is a deleterious mutation which is influencing the function of a protein, then that will be disfavored. So it will lost with time. Now proteins folding impact on the function. Now how does protein folding influence the function, that we'll see. So protein folding will determine the function of the protein. If there is misfolding or if there are some mutations in the surface of the protein, then it might lead to aggregation or it might lead to failed or missed interactions. So as we all know that proteins in our bodies, they interact with other proteins. This is how the machinery is working. Protein interactions, they are working in different bodily processes. So if those residues which are involved in these interactions, if they are mutated, then those interactions will also fail, and the functionality which are brought about by that interaction, that will also get failed. So loss of native interactions. So these are different things which can be influenced by the surface mutations. So properly folded protein, it is involved in the functional interactions. So let's consider this example. We have an unfolded protein chain. If there is mutation in this chain, then it will also bring about the change in the folding. So now if there is misfolding, then it will lead to aggregation, and which will further our protein function will be lost because the protein is getting aggregated. And if there is surface change due to this mutation, then the mutated protein will be formed. Now the surface change can be of different types. For example, it might lead to loss of interaction, but at the same time it can also lead to misinteraction. That means some newer interaction is now happening. And then this further new interaction, it can also have two possibilities. One is that this misinteraction or this new interaction, it is leading to some loss of function, or it might produce some new function. So based on those two effects, we can decide whether to consider this mutated protein or whether to not consider during the engineering. And then it might not have any effect. So there are certain mutations which are neutral. So because in evolution, different mutations are already taking place. So irrespective they are harmful or they are beneficial, they are still taking place. So they comes under the neutral evolution process, which do not have generally any effect on the functionality of the protein. So in this case, the protein which will be produced, it will be functional and it will get properly folded. And then other proteins, no interaction, and it might have interaction partner. So that's why it is bringing about the function. So such mutations, they are favored, they are not lost. And now if there is defect in the protein folding, then what are the consequences of the protein folding defect? So first one is aggregation and fibrillation. So in Alzheimer's disease and many neurodegenerative disorders, this is generally observed that the proteins start getting aggregated. Then misfolding and degradation. So in certain scenarios, if a mutation has happened, protein is not getting properly folded, then it will get degraded by the machinery of the cell, in which the protein will be entirely chopped off. Or mistrafficking. So if protein is not properly folded and if has a signaling region towards its N-terminal region, so in such scenarios, if there is some change, then it might get mistrafficked. It might not move from nucleus to cytoplasm, or it might not move across different tissues which it might have moved previously. So there could be mistrafficking. Or destabilization. So protein can also get destabilized and ultimately it will be degraded by different proteases. Change in biological activity. So for example, we have an enzyme which catalyzes some reaction, but now because of mutation its fold get defected. So in such case, its biological activity will change. It might not catalyze the process with the same efficiency. It might lose its catalytic properties, or it might slow down its its catalytic factor will be less. Then comes PCR and Sanger sequencing. So how we can increase or how we can amplify the DNA sequences. So PCR is a method which is the polymerase chain reaction. So because if we want to increase the yield of the protein, if we want to design a protein, then we start with the nucleotides or the DNA. So that's why we are moving from PCR till the protein. So in the protein engineering, first step will be of the synthesis of the nucleotides. So in PCR, we amplify the DNA sequences. If we have a small sample, then we can make multiple copies through PCR. And there are three main steps of PCR, which is denaturation, annealing and extension. So in denaturation, we heat the DNA at 95 degrees centigrade. So at high temperature, our DNA will get denatured into two individual strands. And after this breakdown, because in the PCR we provide the DNA sample, we have primers, because we know that for the DNA synthesis, it cannot initiate the DNA synthesis, the DNA polymerase, it cannot initiate the synthesis. So we require the free 3' OH group, which is provided by the DNA, the primers which are used in the this PCR. So primers are present, nucleotides are present, because after denaturation we will synthesize the new daughter strand. So for that synthesis we require these nucleotides, which are the DNTPs. Then we have tag polymerase. So normally when we study replication in which the DNA is getting duplicated, so in that we have seen the enzyme DNA polymerase, which synthesizes the daughter strand. But in PCR we specifically use tag polymerase, and the reason is that in PCR we are exposing our DNA to high temperature conditions. So DNA polymerase will get degraded, because proteins in the high temperature conditions, they get coagulated, or they might get denatured. So in such scenarios we need an enzyme or a protein which is highly stable at the high temperature conditions. So tag polymerase, which is obtained from Thermus aquaticus, this is thermostable enzyme. So that's why we use this enzyme. And then we use buffer, because in our cell our DNA is present in the liquid condition or in the aqueous environment. So to replicate the same environment we use the buffer. And then we have a PCR tube in which the entire process of this DNA synthesis is taking place. And this is a thermal cycler, which is a machine in which the PCR process takes place, which provides efficient conditions. So in PCR cycle, first step is denaturation, in which our double-stranded DNA breaks into two single strands. Now after denaturation we performs annealing. So annealing means, in this case this happens at the temperature condition of 55 degrees centigrade. So from this high temperature we reduce the temperature. Now the primers will bind. So annealing means binding of another complementary base pairs to the DNA. So this binding is known as annealing. So in which primers will bind. Now as soon as the primer binds, tag polymerase will come and it will start adding the DNTPs. So it will extend the daughter strands from both the directions. And then to allow the combination of different DNTPs, we further need to increase the temperature, so which is at 72 degrees centigrade, which is the responsible for the synthesis of the new strand, in which extension will take place. So in the annealing step, primer will bind, and in the extension step, the extension will take place. So now we have started with one strand of DNA and we have moved to two DNA strands. Similarly, multiple cycles are run parallelly, and in this way we can increase the number by 2n. So 2 raised to power n. So depending on number of cycles which are happening, the amount of DNA molecules will also get increased. And application is that it is used for cloning, mutagenesis, because the DNTPs which we are adding, so we can also bring about certain mutations in the DNA. In the PCR only we can decide what nucleotide to add. So we can induce certain mutations during this process. Then diagnostics. So we all know that in the different case studies or investigations we use PCR, because we have a minute amount of samples, so we can increase its quantity and then we can perform different diagnostics on those samples, preparing DNA for protein expression. So these are some of the applications of PCR. Now next step is DNA sequencing. So now you have obtained a DNA sequence. Now you also need to sequence. For example, if you have induced certain mutations, then to check it, that whether that what is the sequence, whether those mutations have got into the sequence or not. So for that we need to perform sequencing, and one such method is Sanger sequencing, which is the older method of sequencing. Now there are next generation sequencing methods which already have come into picture. So this Sanger sequencing, this is the method which uses the concept of chain termination. In this it uses DDNTPs, which is di-deoxy-NTPs. So normally in our DNA it is the deoxyribonucleic acid, in which the 2' OH group is missing. Instead of 2' OH we only have H, but it has in DNA we have 3' OH group, which is responsible for the extension during this process. So that 3' OH group is responsible for a particular DNTP to bind to next DNTP. But in the Sanger sequencing we use DDNTP, so it lacks 3' OH group. So now it will, whenever a DDNTP will bind, it will not allow our DNA to further extend. So with this we can sequence our DNA molecule, which is our query DNA, because wherever it stops, that means the complementary strand or the query DNA, it has the nucleotide which is complementary to that particular nucleotide where the chain has terminated. So this Sanger sequencing, it is based on the sequencing by synthesis method. That means the query DNA, the query strand, for that it is synthesizing a new strand. So these types of methods are known as sequencing by synthesis methods, where a complementary strand is getting synthesized. So in ddNTP, it is a modified nucleotide which will stop the DNA synthesis whenever it get incorporated. So in this way, multiple fragments will be generated which are complementary to our template DNA or the DNA which we are observing. So we can watch at the signals that where these peaks are obtained. So with that we can figure out, firstly we'll see this particular signal peaks, because each of these ddNTPs, they are fluorescently labeled, so they have unique fluorescent signals with which we will be able to know that what type of ddNTP it is, whether it is DDA-ATP, DDGTP, DDTTP or DDCTP. So in that way we will be able to sequence the the query DNA which will be the complementary to these peaks. So I will not go into the detail of this, how the entire sequencing is done, but this is just to give you an overview that how we can use Sanger sequencing and what is the purpose behind this. So application is that it confirms the gene sequence and mutation in the protein engineering workflows, because in protein engineering we are trying to engineer a protein to bring about the new functionality. So one way in which we change the function is by bringing about the mutation, and to verify that whether in the PCR the mutation which we induce, whether it happened or not. So to verify this we need to perform sequencing. So now what can be engineered in proteins, that what changes we can bring in the protein. So first thing is folding, that is structural change. So thermodynamic stability, for example we already have a protein and we want to increase its stability, so that can be brought about by engineering. So engineering involves shifting the equilibrium between the native and the folded protein states. So if for example we have a certain protein which is now in this folded state, so we can make some changes so that it moves down the energy landscape and it gets into a native like conformational state. So this happens when we are designing new proteins. So in that we can thermodynamically make it stable. So aim is to favor stable and functional conformations, because more the stability of the protein, more it will be better, its functionality will be better and it will be most stable. And then second thing is environmental stability. So that means proteins can be engineered to tolerate various temperatures, pH, solvents, salts and detergents. So as we know that different proteins are getting engineered these days. So such that because different organisms, let's take an example of the prokaryotes which used to exist in the thermophilic conditions, in the high temperature conditions. So now what scientists are doing, they are collecting their proteins and they are studying that what are the structural changes or what are the proteins, how they are tolerating such extreme environments. Because if we can understand the reason behind those adaptations or those conditions, then we can incorporate such proteins into our organisms and make them thermally stable. So we can design such proteins also, such enzymes also which are thermally stable, so by studying those proteins. So we can take inspiration from the primitive organisms, or we can study those things by we can also increase the thermal stability of the protein by making certain mutations and then studying. But that will take a lot of time if we do not know the already available thermophilic sequences. So environmental stability means we design a protein in such a way that it can tolerate different temperature conditions, different pH conditions, and it can be stable in different solvents, salts and detergents. So mostly these kind of engineering is done to make certain crops or certain plants stable at different conditions, so that we can increase their yield. And then comes function. So we can also change the function, we can engineer the function, such as binding interactions. So for example, we have some protein, we can change certain residues in it, we can use the rational protein design methods to bring about certain mutations and then check that whether this mutation is bringing any change in the interaction of the protein. So in that way binding interactions can be improved, and if we have a ligand of our choice, then we can also make the changes in the protein such that it interacts with the ligand of our choice. So this will help in the designing of the drugs, because by looking at certain target proteins, we can design our drugs in such a way that they can now bind to the target proteins or vice versa. So that comes under binding interaction changes. Then catalysis. So there are different enzymes. For example, if from the real world we have to consider certain cases, so we have different detergents or different washing liquids. So they are normally the enzymes which are interacting with the dirt molecules and they are removing the dirt. So these reactions are the catalytic reactions. So now we can improve this catalysis. So now there are certain enzymes which perform this reaction quite quick. So those detergents will be cleaning the clothes in few seconds, whereas the others might take long. So this activity, this catalytic activity, this can be improved. So enhance transition state binding and increase catalytic efficiency. So this is one of the example that how the activity is increasing from this particular state to this state by just making few changes in the protein. So it requires understanding the enzyme's catalytic mechanism. And for each enzyme, if we know what is the catalytic mechanism, then we can make certain changes, because there are certain sites in the enzyme, such as its binding site or its allosteric site. So those, if we make changes in the binding site, then only we can influence its activity. If we make some change in the random site, then it might not increase the catalytic activity. So for making any change in the protein, first we need to understand that it is working on which mechanisms, because if we know which mechanism it is working on, then only we can design the change and then we can influence its functionality. So the key factors which are involved in the protein stability, they are different interactions. So whenever we are designing or we are making new proteins, then we need to keep these things in mind, such that the interactions which are responsible for a particular folding of a protein or particular structure of a protein, so they must be maintained. If there is any change in these interactions, then it will lead to change in the structure of the protein, which might lead to change in the function of the protein. So different interactions which are involved are hydrophobic interactions, which are generally observed in the core of the protein, because these residues are hydrophobic. So in our body, the protein is present in the aqueous environment, so hydrophobic residues will be present towards the core of the protein. They will not be exposed. So they drives the folding, proper folding, because these hydrophobic residues, they will tend to repel water and they will quickly get folded. So they minimize the exposure to water, and these are the hydrophobic interactions. So there are certain amino acids which are hydrophobic in nature, such as leucine, valine, isoleucine. So they are normally observed in the protein cores. Then there are electrostatic interactions which are also responsible for the stability. So electrostatic, as the name suggests, so it involves the salt bridges or the ionic bonds, and it is observed between the oppositely charged amino acids. So in this case, you can see these are the electrostatic interactions or the ionic interactions, so which are taking place between two oppositely charged groups. So one is the positively charged amino acid with the amino group, and this is the negatively charged amino acid with the carbonyl group. So this type of interactions is possible if we have two oppositely charged amino acids. So whenever you are bringing about the mutation in the protein, then you also have to see that what kind of interaction you are influencing. Then comes hydrogen bonds, which are responsible for the secondary as well as tertiary structure, because in the alpha helices we observe this hydrogen bond between I and I+4 residue. So in this way they are also very important. So if there is loss of a hydrogen bond, then the secondary structure will get collapsed, and ultimately our tertiary structure will also get influenced. Then comes disulfide bonds. So these bonds are observed between the cysteine residues, which contains the SH2 group. So that terminal SH2 group is responsible behind the disulfide bond, and only this one residue is responsible for these bridges. Yeah, so this is the disulfide bond. And then metal bindings. So there are different metals which can also coordinate with the protein. So there are certain proteins which has the metal interactions. So in that way, these metal interactions can stabilize the protein structure. For example, hemoglobin, which interacts with iron. So in this way, so now there are certain changes which we can bring to make our structure more stable, such as X to proline mutations. So for example, we have some X amino acid residue, and if we change it to proline, then in such scenarios, it can restrict the backbone flexibility, because we know proline has this chain which is not that much flexible, which is rigid. So which reduce the entropy of unfolded state. So that's why it will promote the proper folding. So to improve the proper protein folding, these mutations can be brought about. Next thing is alpha helix dipole stabilization. So dipole moment in helices, it can be stabilized by placing opposite charged N and C termini. So in that way we can further stabilize our protein. And the next thing is So this is the representation that the glutamate and lysine, they are placed, so they are oppositely charged. So what they will do, they will stabilize the alpha helix. And now introduction of salt bridges. So if we strategically place these salt bridges, then they will also further enhance the stability, especially at the helix and in the flexible regions. So this is about the protein stability, and we have seen that why this stability is important, because it governs the structure of the protein. So in protein engineering, our aim is to make new functionality to the protein. So for that we must keep these points in mind, that the protein which we engineer, that should be stable enough to perform better functions. So different strategies for the protein design. So when we are designing protein, then different things can be done, different ways we can achieve. So for example, if we have a crystal structure, we know the amino acid sequence, and we also have the functional data available with us, then we can go for rational design. So rational design, that means we have something in our mind, we already have some data in our hand. So in that case, we have structure, we design in silico different proteins. So what we do is, with already available structure, we make certain changes such as site specific mutagenesis. So already available protein sequences available, we make certain mutations in certain regions. So in this case, we have a crystal structure, so we have a DNA sequence in which we will make certain mutations, but this is in silico, and to make it in real, we will use different cloning methods. So that we will see later when we will look into the detail of rational design. But here, just keep this thing in mind, that site specific mutagenesis, it will happen at the level of DNA, then only it will come at the level of protein further, when DNA will get replicated and transcribed and translated. So site specific mutagenesis means there is a specific site where we are doing the mutation, and then protein expression will be observed or will be predicted. Simply in the case of this rational design, we will perform different thermal stability assays. So we know, in the previous session we have seen, there are different MD simulation methods in which we just simulate the real world conditions. So in rational design, what we do is, by bringing those mutations, we simulate those conditions, and then we further verify it using different biochemical tests. But in this design method, we are simply doing it in the in silico conditions. So thermal stability is judged. If it is passing the criteria which we have decided for the protein design, then the protein which is thermostable is achieved. But if it still lacks, then we further need to perform certain improvements, and we again redesign it. We bring about mutation at some other region, or we see that why it is failing. So this comes under rational design. And the second thing is that if we do not have any of these informations, or for example, if we want to design an entirely new protein, so that comes under directed evolution, in which what we do is, we bring about the random mutagenesis, or we perform the gene shuffling. So from different gene shuffling, by making certain changes without any pre-existing data in our hand, we make those changes randomly at different sites, or combining different genes to make a new protein. So we screen it and select it from a large library, and then if it passes the criteria, then it will give us a thermostable protein. If it does not pass, then we further again restart with the process. We perform high throughput screening, and then we verify it. So then in the middle, we have a semi-rational design, which combines the information from the rational design and takes into consideration the directed evolution as well. So in this case, we target randomization based on the structural knowledge. So in this, we were performing random mutagenesis, whereas in the rational design, we are performing site-specific mutagenesis, because we already have the information of different sites which can give us better results. So in the semi-rational, we are taking help of the structure, but the mutations which we are doing is the random mutation. So in this way, it is adding the concept from both of these design methods. Then comes site saturation mutagenesis. So this is a type of mutagenesis in which at each site we mutate it into 20 different types of residues one by one, and then we check that how it is bringing about the change. So this screening and selection of the variants is done, and ultimately if that satisfy the criteria, then it will give us a thermostable protein. So thermostable protein was our ultimate aim to achieve, and these are different strategies with which we are moving towards that aim. Now one by one we'll go through all of these methods. So starting with the rational protein design. So it predicts the protein sequence that will fold into a desired structures, and these sequences are validated by peptide synthesis, site-directed mutagenesis, and artificial gene synthesis. So now we have a computer-aided design. We perform site-directed mutagenesis. So individual mutated gene will be obtained. So for this we use the recombinant DNA technology. So after using the computer-aided design methods, we will see that what are the different things which can be done. We shortlist different mutations, and now after shortlisting it, we perform site-directed mutagenesis in the real system. So in which we can use different vector systems. So in this we will be using the recombinant DNA technology methods. So in the next slides we'll see those things. So we bring about the mutation in an individual gene, we will transform it. So transformation, that means that you have a vector in which you have gene of interest, and with the mutation which you wanted, now you move this vector into the live organism such as E. coli or some other bacteria, where the plasmid will get replicated into multiple copies. So that step where you incorporate the vector into the host is known as transformation, and then protein will get expressed. So first of all, the plasmid will convert into multiple copies in the bacteria, and then from all those copies protein will start its expression. So protein will be expressed in the organism in which you are expressing, and then you will purify that protein, and after purification you will check it. You will perform biochemical testing, you will see whether it is thermostable, and depending on your aim you will see multiple things, and if it passes that criteria, then you are present with the improved protein, and if not, then you will again start the process again. So advantages in the rational design is that it is inexpensive, because you already know the pros and cons of the method, you already know which site to mutate. So that site is inexpensive, you do not have to perform all such random mutations and then check or verify it. Then it is technically simple, because information is already in hand. Site-directed mutagenesis is well established. So these are some of the advantages. Now the limitation is that it requires a detailed structure knowledge, but in most of the cases for all the proteins we do not have structural knowledge. So in such scenarios, rational design could not be possible. Then structural data is often limited or static. So in many scenarios, the structural information we have from X-ray crystallography or some other methods, that is static information, and we do not know that in reality when we are performing these experiments in the in vivo conditions, and whether that structure is exactly that way, or it means there are certain alternative forms which can exist for a particular protein. So there are certain limitations, and then predicting effects of mutation is also challenging. So these are some of the limitations of rational protein design. Now we'll see that what is the target structure. So in the rational design, how we begin. So target structure is the protein function, it depends on the structure. So rational protein design, it leverages this to design the proteins with the desired functions, because different studies have shown that a primary sequence of amino acids determines its structure, and different proteins which have similar structures, they have similar functions. So keeping this hypothesis in mind, what a scientist does is that they thinks that if particular protein has this significant structure and it performs certain function, then we try to make changes in a protein in such a way that it also adopts that particular structure, so that functions can be reproduced. So target structure and fold must be known beforehand in rational design. So we must know that what should be the target structure. If we already know that a thermostable enzyme has this particular structure, so the protein on which we are working on, so we can determine it, we can decide that what kind of amino acid changes we can make in that protein, so that we can reach to our target structure. And then it is usually based on a known protein structure. So if we do not have a template, so template means that already available protein structure. So if we do not have template, then we cannot proceed with our rational protein design. So this is the representation that how the rational protein design works. So we have a sequence. Now we can perform different types of analysis, such as template based, ab initio, or hybrid approaches or refinements. So in this sequence we can make many changes, and then we can get certain structure. So this we already have a set of protein structures and sequences. So based on that we make some changes. So these mutations are not random mutations, they are site specific mutations based on the knowledge of existing literature. So by making those changes, we see how the structure is getting influenced. Then we perform molecular dynamics and docking and binding free energy calculations. So if our aim is to design a drug, then this structure is a target structure. So for that drug, whether it will bind effectively or not, we will perform the docking. So docking means that you are looking for the interactions between a protein and a ligand. So that comes under docking. And in molecular dynamics, you see that how your protein is getting folded and what is the actual conformation of the protein. We can also perform binding free energy calculations. So this will tell you the function of the protein. So these are some of the analysis which can be done. Now, activating or repressing the ligand or protein binding, post-translational modifications can be studied at the functional level. So in rational design, evolutionary methods, discrete and continuous rotamer optimization, all these things can be done, and ultimately we can study the evolution of the protein. So the mutations of receptors, ligands, which are responsible for the gain of function, stability, binding affinity, or enzymatic activity can be studied. So this is how the rational design is done, in which we already have the information in our hand, and these are also based on the template based methods. So template is the sequence which is already available in our database or in our set of information, and now the query with which we are working, so we are just aligning the template with the query so that we can get the desired properties in our query as that of the template based structure. And now sequence space. So sequence space means the amino acids which are present in the protein. So proteins can be redesigned using the non-sequence plus structure, so which is known as protein redesign, which we have seen in the rational design technique, or we can also design these proteins from scratch. So that type of method is known as de novo design. When you are not using any template information or any available protein information, so you are starting from this scratch from any random amino acids, so that is known as de novo. When the sequence is unknown, structure is unknown, you combine different secondary structures into different you combine different secondary structures and you make them into different rotations. You perform different rotations and different changes in that, and you join certain backbone molecules to it. So this will lead to a protein sequence which is novel protein sequence, or which is a newer protein sequence. So that type of design of a protein is known as de novo, where you start from scratch. So in protein redesign, most residues remain wild type, only a few mutated for functional changes. So when we talk about the rational protein design, it is known as protein redesign. When you already have a protein and you are simply redesigning it, you are not generating a new protein. So in this, entire protein is wild type and there are only specific sites where you are making mutations to get certain functions. But in de novo design, the entire sequence is new and you do not have any prior knowledge of the sequence. Then comes energy function in protein design. So when you are moving with the rational design, then you also need to see that whether if you have brought about certain mutation in the protein, then you also need to see that whether it is stable or not. Although you have brought about that mutation, but what if it is not stable and it get degraded? So for that we need to find out the energy function. So importance is that the energy function, it is crucial for distinguishing the sequences that will fold into a desired structure from those favoring the competing low energy states. So we must be able to score and rank the sequences based on how well they fold into a target structure. So for example, if we consider this and we have the backbone and the side chain torsional preferences, then we will see that whether after bringing about some change, whether the conformation which the protein is adopting, whether it is stable or whether it is unfavorable. And these are the energy landscapes. So in some cases what can happen is that your protein might get trapped in the local minima or lower or the slightly higher energy state, which is not the native energy state. So we need to make sure that the protein which we are obtaining after the engineering, that should be in the native state or that should be stable enough. So for which we need to perform these energy function calculations, and requirement is that the energy function which we are using, so that must be accurate, that must reflect the realistic physical and chemical behaviors. So when we talk about this energy function, so we have seen in the previous session also that MD simulations, they try to find out what is the energy of a protein, in which we use different force fields. So protein have different residues, different atoms which are interacting with one another. So all those interactions, they have some potential energy in them, and in the protein we figure out that what is the overall energy of the protein, which will determine that whether the protein will be stable or it is unstable. So in that way, whenever we are trying to identify this energy function, then the force fields which we are using, that must be accurate, efficient, that means they must be computationally tractable, and they must handle the computational challenges of evaluating many conformations during the design. So the force fields or different things which we are studying, then they should not be very exhaustive that it takes a lot of time. So this thing and lot of memory of the computer, so we need to make a balance between all these things. So the challenges is that balancing accuracy versus computational simplicity, it is tough, because if we want to get accurate analysis or accurate protein energy function, then we have to perform multiple calculations. That will become computationally challenging. So we need to make a trade-off between both the properties. Molecular dynamic simulations, they are accurate, but they are too slow for the large scale design tasks, and they are also impractical for many sequences. So if you have a very large protein structure, then it will take a lot of time in molecular dynamic simulations. So there are other methods also with which you can study the energy landscape of a protein, so which we will move to if we have a very large protein, or if we have a large scale design task. So now what are the different challenges for the effective design energy functions? So first challenge is, before moving to challenge, we have water in our protein, because in the in vivo state, protein is surrounded by the water molecules. So when we are performing MD simulations, then also we have to simulate the protein with water molecules, because we know protein have means it has the interactions between different residues, but at the same time it is also interacting with the solvent in which it is present, and that is water. So while calculating the energy, we also have to take care of the water molecules which are surrounding it, because they will also contribute to some type of energy, which we have seen in the previous session, that what is the solvation free energy. So that energy also comes into picture. So water molecules, they are very dominant molecular environment for the protein, and they are also key drivers for the protein structure and its stability. And protein water interactions must be accurately modeled when we are designing these proteins. So the challenges is that we have a high number of water molecules which interact with the protein at any time. So each water molecule has many degrees of freedom. That means it can move in any dimensional, any directions. So that means it has many degrees of freedom and possible interaction partners. So modeling all water molecules explicitly is computationally infeasible. And this also we have seen, that if we model the individual water molecules, then it will take a lot of time, which is the explicit solvation models. So in MD simulations, we have also looked into different solvation models, in which we have seen explicit and implicit solvation models. So in explicit solvation models, it calculates the energy interactions of each water molecule with our protein, but this will take a lot of time because we have multiple such individual water molecules. So what we normally do while we model, we consider each of these water molecule as a continuum, as a medium, that each one is contributing the same amount to this protein, so that we can quickly perform this calculations. So that is known as continuum model or the implicit solvation model. So most protein design treatments or design programs, they treat water as a continuum, capture the hydrophobic effects and the solvation polarization, does not model individual water molecules. But the problem will arise if we have some individual water molecules play critical structural role, especially in the protein cores at protein or protein ligand interfaces. So these are the sites where the individual water molecules can play some role. So in that case, if we design it or if we study or if we model it as a continuum, then we will not be able to get into that interaction. So in such cases, we can go for the micro solvation models, in which instead of considering all individual water molecules, we'll consider few of the individual water molecules which are responsible for the interactions. So we add specific water molecules to the rotamers to account for the structural waters, and other water molecules, they are considered to be a continuum. So this is the method in which we are using the implicit along with the individual water molecules. So this is used in the cases where this water molecules, they have some structural roles. So this will take the time slightly higher than the implicit models, but it will be accurate model. Now next thing is, after modeling all these energy functions, so the next thing is the combinatorial complexity in the protein design. So now when we are designing the proteins, then there is a possibility that they can get folded into a multiple protein structures. So there is exponential growth of candidate sequences. So the number of possible protein sequences, it increases exponentially with the number of residues. So if we have more number of residues, then the possible structures will also increase. So even when side chain conformations are limited to a few rotamers, the number of conformations per sequence also grows exponentially. So larger the sequence, larger the conformations which are possible. Even if we have a Ramachandran plot, but still possibility is still very high. So now these bring about the challenge that although you are making certain mutations, but you cannot be 100% sure that what a structure your protein can adopt, because there are still different possibilities, different structural alternate structures which are possible for the protein of your interest. So this depicts that we have different compact protein structures, low energy folds. So among them, what structure will our protein adopt, that is still a challenge. So there are different optimization strategies in protein design in which best sequence focus. So if your goal is to find only the best sequence, then the algorithm needs the lowest energy conformation of each candidate sequence. Amino acid identities can be abstracted and all rotamers are treated equally during the conformational search. So if our aim is to only obtain the structure which is the lowest energy one, there are alternate structures which are possible, which might vary in their energy landscape, and their difference in their energy might be very low, but you might get confused or there is a challenge. So in that way, to resolve this, many people what they does is they from them to improve or to fasten up the selection criteria, they select the one which has the lowest energy. So how this process is done? So you perform energy based optimization models, because in protein design, because you are designing the protein, so we have different amino acids which are present. So we see that what are the different rotamers. Rotamers means by rotating what are the different forms of that particular residue can exist. So for example, if we have Ri, which is the rotamer at position I in the protein chain, and Eri is the internal energy of the rotamer I, because our aim is to find the energy of entire protein. So in this way, we are finding for the one particular amino acid which is I, we are finding its internal energy, and that particular amino acid, it is interacting with other amino acids. So we find that what is the interaction energy between our residue I and all other residues, that means J. So total energy is given by minimum ET, that means our aim is to get the minimum total energy, which will be equals to summation of starting with I, because we are considering I as the current residue. So EIRI plus EIRI means energy of that particular residue I, rotamer, and then plus summation of all its interaction energies with all such J residues, and such that we are not considering I equals to J. That means the residue itself or the amino acid itself, so its energy with itself is not calculated, its interaction with all other residues is calculated. So it should be minimum. So that will tell us what is the total energy, and this will be repeated for each residue. And overall, after summing all these energy functions, the one which gives the minimum energy will be selected. Yeah, so this is how energy based optimization is done. Now there are different protein design algorithms which exist, which helps you to find out that which of these conformations should we proceed with, which one of these structure is the lowest energy conformation. So different algorithms, one among them is exact algorithm, and the example of exact algorithm is dead end elimination. Now we'll see what is this dead end elimination method. So it eliminates the suboptimal rotamers without losing the global minimum energy conformation, because we know there are certain local minima which also exist. So when we are comparing different energies, then dead end elimination help us to get that particular conformation which has the lowest of the energies. So for example, for residue I, we will eliminate that rotamer if the energy of this rotamer, which we have seen in this particular equation. So now this one is for the single I. So energy of residue I plus its interaction with all such residues, if we add all the interaction energies, then if they are greater than the energy of I plus summation J max E, so this is the rotamer R prime. Now this I residue, it can be in different, by changing its angle, it can exist in different rotameric forms. So for example, if the energy of rotameric form Ri is greater than the energy which we obtain from R prime I, then in such case the Ri rotamer form is removed or it is ignored. We eliminate this Ri because our global aim is to get the minimum of the energy. So Ri prime is the another rotamer of the same residue which will be selected because it has the lowest energy than the Ri form. So if the worst case interaction energy of another rotamer is better than the best case of the current one, then we will eliminate the current rotamer, because in the current rotamer we were looking for the minimum of the energy among those interactions, whereas in the case of R prime I we were looking for the max of energy. That means max energy is the one which is the worst case. So in this, if for the current rotameric form, if its best energy is greater than the worst of the energy, then we will ignore this kind of rotameric form. So which will help us to get that particular rotameric form which will give us the least of the energy or the lowest energy, which will be the most stable protein structure. So this one is the exact algorithm, and the pros of this algorithm is that it guarantees you the optimal solutions based on the protein design models, but the limitation is that it do not guarantee you runtime efficiency, because if we are looking for multiple comparisons among these rotameric forms, then it will take a lot of time. So it is not that much efficient. It can fail in experimental validations if the energy function or model assumptions are flawed. So in these calculations, if there are certain flaws, then in experimentals you won't be able to reproduce the same results. So this can also fail. And the sources of errors could be inaccurate energy functions. So the energy functions which you have considered, they might, if they are inaccurate, then it will complicate your results. Limited sequence space or insufficient target structure flexibility. So these are some of the errors which can be observed in the exact algorithms while you are calculating for the energy. And when you are comparing different conformations, then you might pick up that conformation which is not the right one due to the error in these function values. Now the second approach is the heuristic algorithms, which involves the Monte Carlo methods. So this one is quite fast, but the problem is that it does not guarantee that whether it will find the optimal solution, and it is useful when you have the time constraint. When you have to perform this identification of proper conformation quickly, then you go for heuristic algorithms in which you select a move. So select move means we have different rotameric forms of particular residues. So we select that move, rotation, transition. If that move is valid, then we select it, we compute the energy, we perform metropolis calculations. If its energy is less than what we are expecting for the another rotation, then we will accept it, otherwise we will reject it. And similarly, if the move is not valid, then we will reject it and again start with the another rotation angle or another move. So simply we have the sequence in our hand and we are just trying to change its different rotations. We are making the change in the torsion angles so that the structure which we get must be highly stable, and it should be in the low energy conformation state. So this is the heuristic based algorithm, and it might get trapped in the local minima. So that is the limitation, that it does not guarantee you the structure which you get, it will be the optimal one. Now the third method is the Rosetta design or OS spray. So which is a sophisticated method because it is combining the information from both these algorithms. This is the improvement. So that involves the Rosetta design and OS spray. So in Rosetta, it uses sophisticated energy functions. It employs the Monte Carlo with backbone flexibility for efficient conformational search. So in Monte Carlo, it means we randomly move in the energy landscape such that high energy move is also favored by increasing the temperature. So in that way, we are also incorporating the backbone flexibility so that the conformational search become efficient. Then in OS spray, we extend the DE, which is dead end elimination, with the continuous backbone and side chain movement modeling. So at the same time, because in these rotameric forms, we are limited to only the phi psi angles. But in this, we will also be looking into the side chain movement as well. So chi will also be come into picture. So ultimately the results which will be obtained, they will be more better, more confident enough for the particular protein structure stability. So till now we have covered what are the different aspects, what are the things which we need to keep in mind while designing the proteins, and how to determine that the protein structure which you have obtained, whether it is in the native state or in the low energy state conformation or not. So now we will move to the second approach with which you can design the protein, which is also known as the directed mutagenesis or directed evolution method. So before moving into that, we'll see what is mutagenesis. So mutagenesis means when you bring about the change in the DNA sequence, you are making the mutation. So mutagenesis means giving rise to mutation in the DNA sequence, and most of the times we study the point mutations. That means we make change of only a single nucleotide. So that type of mutation is known as point mutation, where only a single nucleotide base is changed. It could either be inserted, it could be deleted, and changed means it is substituted with some other amino acid residue. And there are different types of point mutations such as substitutions, insertions, deletions. And along with this, what are the consequences of these point mutations? So based on that, point mutations can be, for example, if we do not have any mutation, then this is the DNA, this is the mRNA, and it gives rise to a lysine amino acid. So silent mutations are the one in which the nucleotide is changed into another nucleotide, but the amino acid which it is forming is the same. So when you are observing the protein, it appears that there is no mutation. So these type of mutations are known as silent mutations, which are taking place but their effect is getting silenced. Next is nonsense. That means the mutation is such that it gives rise to the stop codon, and your protein which is getting synthesized, it will get terminated because now stop codon will come into that site. So instead of lysine, we have a stop codon, so protein will terminate at this point. Now the third type of point mutation is missense, which could be both conservative and non-conservative. So missense means the mutation in nucleotide will change the amino acid in the missense mutation, but if that amino acid has the same physicochemical properties, for example, lysine was a basic amino acid which is positively charged, and arginine is also positively charged amino acid. So this means the property is conserved. So this is known as conservative missense mutation. Whereas at the same time, if that particular change means it is changing the amino acid, but the amino acid which is replaced, which is changed, that has different properties. So now it is polar. This is threonine. So if lysine is replaced with threonine, then its property has also changed. So these type of changes are non-conservative. That means this mutation has not conserved the functional information. So till here it has conserved the functional properties or physicochemical properties, but this has lost it. So this is non-conservative missense mutation. Yeah, and this is the codon table. So this decides that which of the three nucleotides will form which kind of amino acid. So if you are interested, you can go through this codon table. And now there are some other types of point mutations such as transitions. So these are the point mutations at the level of nucleotides, and these were the consequences, that what are the results of these mutations. And these are the DNA level, means what are the types, or means what are the things which are changing. So on the basis of that, point mutations can be transitions. Transition means that if you are changing a purine with another purine, or if you are changing a pyrimidine with other pyrimidine, such as C with T or T with C, and A with G and G with A. So these are known as transitions. Transversion means that you are changing a pyrimidine with the purine. So you are changing the type of the nitrogenous base. That type of change is known as transversion. Then comes deletion, in which you are deleting some of the region. That could be either a single nucleotide or more than one nucleotide. So that type is known as deletion. And in this case, due to this deletion, so in this the G is deleted because this is a point mutation. So at one time you are only changing a single residue or single nucleotide position. So in this, G is lost, due to which now previously GGA was there, so it was forming glycine at the second amino acid position, but because G is deleted, so now it will form a codon GAA. So instead of glycine it will form glutamate and followed by valine. So now this has changed the amino acid sequence. So deletion can also bring about the change in the amino acid sequences, and which will ultimately change the properties of that protein, its structure, its function. Insertion means if some new nucleotide get inserted, so that can also change the amino acid sequence of the protein. And this has not just changed one sequence but the entire protein sequence, that multiple residues get changed. So these type of changes, they are known as frame shift mutations, which changes the frame in which the protein was first read. So we will not go into the detail, but this is just an overview that how the mutations can be performed and how they can bring about the change at the level of protein. Now the journal strategy for this directed mutagenesis. So our hypothesis is that natural DNA sequence, it can be modified to improve the functions. Now changes must not disrupt the overall structure. So whenever we are bringing about the mutation, our aim is that the structure should be maintained. It should not lose the structure. And now the new sequence, it should also be similar to the original structure, that it should preserve the function. Along with this, that mutation should also lead to a new functions. So this is the hypothesis. Now the approach is, first is the site directed mutagenesis. Site directed means you perform the targeted point mutations. That means you know that at which site you want to see the mutation, and you ultimately perform that mutation in the gene. You insert it into the vector, you perform transformation, you move that vector into the host, which is E. coli. You express the protein in E. coli and you purify it, and then you check for the activity, stability and structure. This is known as site directed mutagenesis. Now second approach is random mutagenesis. That means instead of targeting one particular site, you are randomly mutating the different sites, and then you are checking that how those mutations are bringing about change in the protein function. So random mutagenesis is done when you do not have much information that which sites should be modified, or when you wanted to explore different mutational effects. So in such scenarios you go for random mutagenesis, and ultimately you will transform it, you will express it, and then you will check that how it is influencing the activity. Either it is a negative mutation or it is improving the mutant. It is stable or it is unstable, and it is improving the activity. For example, if you are considering the enzyme, then you will see that whether its catalytic activity is getting improved or it is getting worse. So based on that, we will figure out that which of these mutation is helpful. And the application is that it helps you to study the function of proteins, DNAs and proteins, and it is very crucial in protein engineering, because in protein engineering, when you are trying to change the function, then you must need to change the protein sequence, because sequences are responsible for the structure and ultimately for the function. So this is the strategy. With the mutation, you can change the protein, you can design the protein with new function. And it also enables you to create the DNA libraries by intentional mutation for functional studies. So in that, by random mutagenesis, you can also see that which of the mutations, how the mutation is influencing the change in the protein functionality. So with this, you can construct a informational data set, which can further help other people who want to study in this realm. And then DNA shuffling is also one of the method in direct evolution. So in which you have different genes, you shuffle them and you recombine them to make a mosaic gene, which is the combination of different parts from different genes. So this is also one type of way in which you can change the functionality of the protein. So instead of bringing mutation, you are simply shuffling the parts of the genes. Now if you want to design completely novel protein, then that comes under de novo protein design. Till we have seen two types of protein designing methods. One was rational protein design, and the next one was the directed mutagenesis or directed evolution. Now third one is de novo protein design, in which you are designing the protein from scratch. You do not have any information about the sequence, about the structure. So with this, what you do is you take different secondary structures which are already available, you idealize them and you combine them in different combinations and you reconstruct a fold. So in this way, you are designing a new protein which was not existing previously, or fragments from different proteins. So in this case, from different proteins, different segments are obtained and the protein is reconstructed. So in this way, to some extent, we are using the information, but for that particular protein, we do not have any such information. So we are making a protein which is completely unique. Now, sequence design and optimization. So when we are combining them, then we also have to do the optimization, and as we have seen that we need to see that the structure which is produced, it should be stable enough. So all those optimizations are done, and if it is not stable, then we will make certain mutations, certain changes, so that we can increase its stability. We can refine it to obtain a low energy sequence which will be giving us the lowest energy conformation, and ultimately we will validate it with the experimental methods. So this is the de novo approach. As it is tunable, it is easier to optimize, and it is also helping us to build the enzymes with the new activities and non-natural amino acids, because we are designing it from scratch. So we can also insert some non-natural amino acids so that it can improve the functionality of that particular protein. But the challenge is that it is complex, because we do not have any information, so it is complex. It's not always guaranteed that the protein fold which you obtain in the end, it will be better, or it could be optimized, or it will be functional in the experimental scenarios as well. So there are many challenges which are associated with it, and it will also take a lot of time and months of iterations. So despite there are advancement in algorithms, but it is a time consuming process. Although it will help you to design the novel proteins, but it is not always guaranteed that what you are thinking in your mind, it will work out or not. Yeah, so this is also the way in which you can, with de novo, you can decide that what function you want to see, what structure you want to see, and what sequence you want to see in your protein. So these are different choices which you can make while designing the proteins using de novo methods. And there are different protein structure prediction methods which are available, such as knowledge based methods. So the technique is that the fold recognition by comparing an unknown sequence to a known 3D protein structure. So for example, if you have a sequence for which the structure is not predicted, then in knowledge based methods, you simply look for a template. You look for a sequence which is most similar to the sequence of your query sequence. So in that way, because why we are looking for template based modeling, because till now we have seen many times that the sequence is responsible for the structure and structure is responsible for the function. So the sequences which are similar in the if two sequences are similar, then they will most likely make a similar structure. So to predict the structure in the knowledge based methods, we look for the sequence which is similar. So we do template based modeling. And there are two types. One is homology modeling and another one is threading. So in homology modeling, we simply look for the sequence similarity. Whereas in threading or fold recognition, we match the sequence to existing fold structures even with low sequence identities. So even if the two sequences do not have high sequence identity, but we'll simply see that what fold it can adopt. So we look for the existing fold structures, and these can be combined. So for certain regions where homology modeling fails, we can do threading. Yeah, so selection of best template is done, and then alignment of template and target sequence. Target sequence is the one for which we are trying to predict the structure. We build a model and we optimize that model, and ultimately we will validate that model. So this will help us to predict the structure for our target sequence. In threading, we select the template fold for the target. So in this, we only look at the folded region. In homology modeling, we model the entire protein. In threading, we only model the folded folds. So alignment of target and template folds. So from entire sequence, we only align the folded folds or that particular region which is involved in the folds. We build the model, we evaluate it, and we validate it. Next method, if we do not have template based information. For example, if we are not able to establish the sequence identity to other sequences, so in that we move to template free modeling, which is the ab initio modeling, which predict the structure using the physics based simulation without any template. So in this, we conformational search using the energy functions. Model selection is done and reconstruction of all atom structures. Structure refinement is done, and ultimately model is validated and that structure is predicted. So the basis is that it simulates physical forces and chemical contacts. So this simply checks that what are the possibilities. If we have these many amino acids, then how they can fold. So in ab initio methods, instead of using any already available information from the template, we simply use the physics formulas and we see that what is the possibility of contacts which are available. Then we simply use energy minimization and molecular dynamic simulations to simulate the structure. Our goal is to find the lowest energy conformation and most likely the native fold. So previously we were looking for the different rotameric conformations. So in this also we will do the same. We will look for different rotamers which are giving rise to the least energy state. So that is our aim, and the advantages is that this works for the novel folds. So there are certain folds which are not already, for which information is not available. So in this case, using ab initio methods, we can predict them. But the disadvantage is that it is only limited to short sequences because it is high computational cost. So if we have a very large residue, then doing all the rotameric combinations and doing that permutation combination thing, that will take a lot of time as well as computational resources. So in such scenarios, ab initio methods are not that good. So now we'll see another tool, which is the ITASR, which is known as Iterative Threading Assembly Refinement. So this is a structure prediction server and it has different stages. First stage is threading. It identifies the template proteins with similar structural motifs using the solved structure databases, and it aligns the query sequence to these templates. So we have an unknown sequence for which we want to we have a sequence for which we do not have a structure available. So to predict its structure, we perform threading. We see in the sequence that whether already available database has the fold information to these regions in the sequence. So we simply align those regions which align to a folded sequence. So we perform multiple threadings. So because there are multiple motifs which are present in a protein sequence, so we model all those motifs. So that is known as threading, in which we are only looking at the folds of the protein. Now after getting different fold information, we perform structural assembly. So we extract the aligned fragments from templates and we construct 3D structure using both aligned regions and ab initio modeling for unaligned parts. So in threading, we will only be able to model the folds, but the region which is left, for that we need to do the ab initio modeling. So we will use ab initio modeling for the unaligned parts. And then next thing is so this is the structure assembly. We'll do clustering and then structure reassembly, then lowest E structure. So that means lowest energy structure is chosen, and then optimizations are done to get the final model. So in this, cluster analysis of predicted model is done, in which different folds are clustered and then we combine them to get that fold which is in the lowest energy state. We will refine the best structure by simulations, and then structure based function annotations are done after TM align search is done, and from the PDB library we look for the structure analogy. We see that whether the structure which we have predicted, either that similar structure is available or not. So that is known as structure based functional annotations, in which we can classify the EC number. For example, if we have an enzyme, then we can look for its EC number. We can look for the GO vocabulary. So GO is gene ontology vocabulary, in which we will see what is the functionality of that particular gene. For example, if the structure of our protein which we have predicted, if it matches to some other protein structure, then we will see that what was the gene of that, or what was the functionality of that protein. So in that way we can annotate its functional information. We can also look for the binding sites. So these are known as functional predictions. So this is how ITASR is helpful in predicting the structure of the protein. And in this LOMETS threading, so LOMETS means local meta threading servers, and it is a critical first step in ITASR pipeline because it uses multiple threading methods to identify the template fragments from known protein structures, which are then used to assemble and refine the accurate 3D models of the target proteins. And LOMETS is providing you the structural blueprints, because for different regions in this sequence, it will tell you what are the different folds which could be mapped from the existing literature. So this is the ITASR method. Now next is the Rosetta structure prediction server. So it includes algorithms for computational modeling and analysis of protein structures, and it also searches for the lowest energy conformation of a protein sequence. So its workflow is that you have an input query sequence. Now you create a library of different fragments which is profile alignment based. Now from them you go for the Monte Carlo fragment assembly. So in this, randomly sample the backbone conformations, and then you perform coarse-grained energy functions in which you will perform certain calculations to generate low resolution models, and then all atom energy functions are calculated in which you apply the physics-based atomic refinements, in which you apply this information to find out that whether the atomic model which you are getting, whether it is in the lowest energy state or not. So you keep on refining it again and again by changing the different rotamers, and then final model is obtained which has the lowest energy state. So this is the Rosetta based structure prediction, in which you are not using any template information. So you have the knowledge based potentials, but you are using the physics based assignments to predict the structure. So the strategy for de novo design. So if you want to design the protein, then you will define the desired structural shape, which could be a platonic ideal, and then design thousands of backbone conformations, add side chains, refine and test folding predictions, and synthesize the best candidates, test them, iterate them, and optimize them. So these are different strategies. So we have already seen that when we want to design the protein de novo, then we use the physics-based methods, because we do not have already available protein sequence. So we simply combine different units and then we refine it. We see whether it satisfies the physical criteria or not. And then other methods are like DNA origami, in which we use the DNA to make different three-dimensional structures, and the main mechanism which is responsible for this three-dimensional structure is the complementary base pairing, because in DNA we know that the base pairing is complementary and it is very specific. That means adenine will only pair with guanine and cytosine will only pair with thymine. So that base pairing, that hydrogen bonding is very specific, so with which we know that which base pairs can connect to one another. So we can have a DNA scaffold in which we can use staples. So staples are also the DNA sequences in which we already know that what are the nucleotides which are present in it, so that we know that where it can bind, because they show specific complementary binding, so with which it can adopt different structural forms by using scaffold DNA and these staples, which can only which will bind to only those regions which contains the complementary nucleotide base pairs. So in DNA origami, we are designing the different three-dimensional structures or two-dimensional structures from the DNA molecule in the nanoscales. And the application is that it can act as a construction material, because we can predict the base pairing, and the inspired efforts to design orthogonal protein pairs, engineered proteins that interact only with the specific partners. So from DNA origami only, different scientists were inspired and they generated the orthogonal protein pairs. Then next is the blob level protein design, in which what we do is we combine protein domains from different proteins and we make a fusion protein. So fusion protein means which has domains from different other proteins, and now each domain has some function. So the protein which we have now designed, it will include the functionality of these different domains. So we will check that whether these two domains, when they are present together in a single protein, then whether both the functions are retained or not. So this is just the representation that these are different domains which are combined in different orders. So now they will have different functionalities. They might increase the hydrophobicity, whereas certain domains, they are non-hydrophobic. So all these things, all these changes, depending on our need, we can use this blob level protein design to make the protein of our desired functionality. And applications are, this is generally used in the sensors, signal transduction pathways, transcription factors, and different therapeutics. Now if we talk about the directed evolution of proteins, so we have already seen that how we can do this directed evolution, in which we either do the site specific mutagenesis or we do the random mutagenesis. We express it in some E. coli or organism where they make multiple copies of that protein. We select it, we verify it, that whether this particular mutation is beneficial or whether it gives rise to a structure which is unstable or not. So we select that mutation and we isolate it, and then further we get the output. So we increase the production of only that particular protein, that particular protein with the mutation which is desired, which will increase the activity. So this is the concept of directed evolution, and the key feature is that this is iterative process. You diversify, you screen, you rescreen. So iterative process in terms of if you are doing random mutagenesis, so then it is iterative. You can tailor enzyme for new environment, new substrate, novel catalytic activities. Now, different molecular insights and library designs and directed evolutions. So, where to perform these mutations? So, if we are considering the enzyme, then normally we bring these mutations either near the active site, because if we change the residue in active site, then it can change the catalytic efficiency. It can increase it or decrease it. So, means these are the sites where the mutations are preferred. Next is mutation on the enzyme surface. So, because we know that a particular fold of the because proteins get folded into a three-dimensional structure, so the surface is the one which is interacting with the aqueous environment. So if we change the residue on the surface, then also it can change the stability. It can either increase the stability if that residue is hydrophilic or polar, but if it is hydrophobic, then it will means reduce the stability of our protein. So in this way, these are the insights, these are the things which we have learned through time, that what are the favorable sites to bring about the mutations. Next was the DNA shuffling, the stemmer strategy, which also we have seen that if we do not know where to perform mutations, then we can also reshuffle the DNA. We can combine different gene units together and we can design a new protein out of it. So step by step is the introduce random mutations in the enzyme gene, insert gene into bacteria, express the mutate enzymes, test that enzyme activity, and then mutate best enzyme again. So you will repeat the cycle till the time you find that mutation which is trying to increase the activity of your enzyme. If that particular mutation is getting you the enzyme which is losing the function, or in which the activity is reduced, then that particular mutation is skipped. That mutation is not kept further, because in this our ultimate aim is to increase the activity of our enzyme, catalytic activity of our enzyme. So in that way we keep on doing mutations till the time we fail or we are not getting any further increase in its catalytic values. So this is the molecular insights and the library design. And now the selection criteria. So how you select that, whether the changes which you have done, whether you should select that mutation or not. So first step is thermostability. So if your goal is to enhance the thermal stability of enzymes, then you will check for the different, you will check it using different assays. You will interleaved heat treatment, you will do interleaved heat treatment, and then you check that whether at that temperature how is the activity. So you compare the wild type and mutant activity. So if at higher temperature you observe the activity, if it is stable, then that means you can proceed with that mutation. You can measure the thermal stability and you can look for the shift in the melting temperature. So melting temperature where your protein get degraded. So till then you can compare melting temperature among wild type and mutant. If your mutant has a greater melting temperature, then that means your mutant is thermally stable. Second thing is you can look for the improved or change in catalytic activity. So for example, if you have the catalytic activity till here, then you will see that if you have made certain mutations, then whether it is leading to increase in the catalytic activity or not. So that also can be governed by looking at the catalytic activity in the real space. If you are doing certain mutations, then you can make a plot of their catalytic activities. So the mutation which is giving rise to or which is increasing the catalytic activity, that mutation will be kept, and other mutations, they will be discarded, they will not be further considered. So if enzyme prefers a natural reaction, it lowers its natural activity first, then redirect evolution efforts to improve the activity for the new reaction. So you keep on doing new reactions so that you can increase its catalytic activity, or you can change its catalytic activity. Then applications of this directed evolution is carotenoid biosynthesis metabolic pathways. So with help of directed evolution, what you can do is that you can design the multi-enzyme pathways, that instead of this is the example of the carotenoid biosynthesis. So instead of going through one pathway to design or to construct or to produce carotenoids, you can use multiple enzymes to produce the carotenoids. So with the help of this multi-enzyme pathway, you will be able to produce multiple such carotenoids with different enzymes. So multiple enzymes can be used, which will help you to enhance the output of beta carotenoid which is produced. So you are not limited to only one enzyme pathway. So this is the advantage of directed evolution. Second advantage is biofuel productions. So The leading biofuel candidate is the recombinant E. coli, which is generally recombined. So there is a problem in this E. coli when it gives rise to biofuel is that there are two pathway enzymes which require NADPH, while E. coli mainly produces NADH during glycolysis. So we do not have enough NADPH because we need to convert NADH to NADPH. So how to use protein engineering such that we can increase the biofuel production? So solution is that to do such a mutagenesis or such a mutation such that it can alter the enzyme, that the enzyme which was previously using NADPH for biofuel production, now we change that enzyme in such a way that its substrate gets changed. So instead of using NADPH, now it could use NADH. So in that way, directed evolution can help you to improve the compatibility of the enzyme with respect to the host metabolism and makes the organism more suitable for biofuel production. So with this, biofuel production can be increased. So the E. coli do not have to convert NADH to NADPH. With NADH only, it can make more and more of biofuel. And then the next application is new chemical bonds. So new chemical bonds can also be introduced by bringing about certain mutations such as carbon silicon bonds, which are prevalent in synthetic chemistry, but they are absent in nature. So natural enzyme do not catalyze these carbon silicon bond formation. So directed evolution can help you to engineer the enzymes to perform this novel chemistry. So with the help of directed evolution, now you will be able to form the bond between the silicon and this carbon. Previously this bond was not catalyzed by any enzyme. So this is also the help or the application of directed evolution, that you can construct new chemical bonds. Then ensure selectability of enzymes. That means with the help of directed evolution, you can also improve the ensure selectivity. That means important for asymmetric catalysis. So evolved enzymes can produce the chiral molecules which have the high enantiomeric purity. So for example, directed evolution on transaminases increase the S selectivity from 65% to 94% for converting beta tetralone to its amino form. So in this way, enantiomeric form selectivity can also be changed. So previously, if it was selecting the another enantiomeric form, so now 65% it's increased to 94%. So most of the times when this protein is in its S form, then it gets selected and it converts to beta tetralone. So this is the application, another application. Now we have seen all the applications. So now, because we have seen the advancement, so machine learning is also has advanced. So with the help of machine learning also, we can improve the protein designing, in which we can perform the protein engineering. So how we can use the machine learning? So when we are using the protein design methods, then the limitation or the bottleneck is that the screening is very resource intensive, because when we are doing random mutagenesis, then how to screen it is a lot of time consuming method. So with the help of machine learning, we can reduce that experimental burden. We can predict which variants are worth testing. So in this, with the help of in silico methods, we can shortlist the variants with which we can further perform the experimental validations, because it is not feasible to perform experimental validation with all the mutant forms. So with the help of machine learning, we have reduced the burden on the experimentalists, or we are reducing the resources which were previously used. And another alternative is the molecular dynamic simulations, which are computationally intensive. So with with the machine learning, we can shortlist them and then we can go for molecular dynamic simulation for those scenarios. And ultimately, after validating with the molecular dynamic simulations, we can go for the experimental validations. So this will reduce the burden on the resource, and this will efficiently help us to design new proteins with better functionality. So advantages is data-driven decisions. So machine learning is entirely based on the data-driven decisions. So if you do not have a data, then machine learning cannot be used. Then it speeds up evolution by avoiding the exhaustive wet lab works or wet lab screening, and it allows the rapid exploration of sequence space versus months of experimentation. Yeah, so this is the example that, for example, if you are doing it experimentally, then maximum times you get the variants which are unimproved. So you are losing the time. But with the machine learning, you can perform the mutagenesis, you have a variant pool, you can screen it, you can make the sequence function pairs based on the available knowledge, and you will design that which sequence can make which function. So the sequences which gives you the better functional output, that particular sequence change you will consider, and you will go with the experimental validation for that particular mutation only. So this is how ML guided selection can increase the chances of getting better outputs in the experimental methods and reducing the efforts. Yeah, so similarly in this also the same thing is being shown, that how with the help of machine learning you can reduce your efforts. And then there are different machine learning model development which are used in protein engineering. So QSAM is one of such model, that is quantitative sequence activity modeling, in which you compare that how the sequence affects the activity, and it is inspired by QSAR and QSPR, in which you know how the sequence or how the structure is influencing the activity versus sequences affecting the property relationships. So these are helpful to predict. So these are ML models which will help you to predict that how the function will get changed with the sequence, so that you can shortlist those mutations which could give you the better results and then proceed with the experimental methods. So one such QSAM algorithm is ProSAR, which fits a linear model using the sequence activity data. We already have a data and we know that which sequence will have, how the sequence is responsible for the activity. So for each sequence we have this data, and then we classify our mutations, that whether that particular mutation will be beneficial, deleterious or neutral, and we will determine that which of the mutations will be beneficial. So those mutations will be considered and performed in the experimental setup. Yeah, so this is the plot, that these are different sequences and these are the properties shown by each sequence. So the predictor will predict the it will tell you that which of the sequence will have what type of output, whether that particular mutation will be beneficial, that means it will increase the activity, or it will decrease the activity. So all these predictions can be done by using this ProSAR algorithm, and the application is that the substitutions identified by these ProSAR can be retained or removed for the next evolution cycle, and it enables you to perform the 4000-fold improvement in the enzyme function using 35 substitutions. Yeah, so these are different libraries which are available and different mutation combinations, and then from all the possible mutations which can be done, you figure out that which of these mutations are beneficial, you keep them, and then those mutations are further selected and then they are checked. So in this way you are kind of improving the functionality of your enzyme by multiple times, because if in experimental setup you need to find out the number of mutations, then it will take a lot of time, but with the help of ML you are simplifying it. You are using the predictors to predict the activity. Next concept is epistasis. So this is a phenomena in which the effect of mutation in one gene depends on the presence or absence of mutation in one or more other genes. So the mutations which you are bringing about in a protein, so protein is not an independent unit. It is interacting with many other proteins. So it is also possible that the mutation at one region can influence the protein at some other place, the protein which is getting expressed some other place. So that is what epistasis tells you, that effect of a mutation in one gene can also depend on the presence or absence of mutation in some other gene. So those two mutations can be dependent on one another. They are determining that whether it will bring about the effect or not. So there are two types of this epistasis. One is negative, another one is positive. So negative means the combined mutations, they worsen the effect of the individual, or the positive epistasis means then the mutation restores or improves the function. So that is known as positive epistasis. And now how they impact the directed evolution. So for example, if we see here, then this green one is, for example, let's consider the example of green fluorescent protein. So if you bring about any mutation and it is also giving a green dot, that means that mutation is not changing the function. But in other scenarios it can change the function, it can lose the function. So GFP is no longer GFP. So this mutation is deleterious mutation. But what can happen, let's consider this case in which the green is mutated to dark gray, in which it has lost its function, but some other protein has some mutation. So due to the epistatic effect, now the function is restored. So in this way, this epistasis is very important to study, because for example, you make the mutation in the one of the protein and you want that it should increase the activity of the enzyme, but experimentally when you validate it, you see that there is no increase in the activity. So now why you did not see any increase? Because the reason could be that that particular protein mutation is also somewhat, some other mutation is lying in other protein which is not done, due to which this mutation is not showing its effect. So those two mutations could be linked. So that's why in directed evolution we must see that whether the mutation which we are bringing, whether it has some epistatic partner which is existing or not. So that is the impact of this particular epistatic thing on the protein mutations. And role of machine learning. So machine learning helps you to navigate the complex epistatic landscapes, because in machine learning you can give the data sets and you can look for the correlations, that which mutations are correlated or which are always observed together, and they are kind of, means these are just the predictions with which you can predict that either two mutations are linked to one another, so that whenever you are designing new proteins, so in that way you can design the mutation in other protein as well to get the desired results. So it is useful in predicting the combinations that might lead to improved protein functions. And then there is a suite of programs which is IPRO, that is the Iterative Protein Redesign and Optimization suite of programs. So its purpose is that it was designed to redesign proteins to enhance or provide the specificity for native or novel substrate cofactors. And the approach is that it repeats the random perturbations of protein structures at different positions. It keep on perturbing it until and unless you get a structure which is in the lower energy cutoff, whose energy is as least as possible. So till that point you will keep on perturbing, you will keep on changing the things in your protein sequence to get the most stable protein conformation. So the capabilities is that you can graft the binding pockets into existing protein scaffold, antibody structure predictions can be done, you can enhance the enzyme activity, structure and energetic assessment can be done for specific mutants. So perturbation means you are changing the protein, you are changing, either doing the change in the rotations or you are changing the mutants. So this comes under the IPRO, in which you redesign the proteins and then verify that how those mutants are linked with the activity or functional properties. So one such case study is the Pore Designer. This is a tool in which helps this redesign the bacterial channel protein, which is OMPF, to reduce the pore size from 1 nanometer, that is 10 angstrom, to sub-nanometer dimensions such as low as 3 angstrom. So how to reduce this pore size? Because if pore is large, then the salt will enter into the that particular bacterial organism. So in this way we are trying to reduce the pore size such that it can reject the protein salt. So how to reduce it? So with the help of designing strategies, there could be site specific mutations. So what kind of mutations were done? So glutamate at 62 position is replaced or substituted with tryptophan 62, and simply arginine is also replaced with tryptophan. So as we all know that tryptophan is the one of the amino acid which is the biggest among all such amino acids, and this is also highly hydrophobic. So with the change or with the mutation of this amino acid, this will make this protein channel to be hydrophobic, and this will reduce the pore size. So with the help of Pore Designer tool, this particular OMPF was studied and the mutation was done and it was observed, and then later on it was also experimentally verified that it leads to reduce in the pore size. So this is one of the advantage, that with the help of protein designing you can also, how you can change the physical properties of the pore size. So outcome is it resulted in the smaller pore lumen and created a more hydrophobic inner pore wall. So this designated pore shows complete salt rejections in biomimetic block polymer matrices. So this is the one of the case study which highlights the advantage. Second case study is of the engineered protein cages for enhanced stability. So in this also protein, previously there were water molecules present between these arginine, that is N. So now these N's were replaced with tryptophan, phenylalanine and lysine. So with this, the relative cage formation and the thermostability values also changed. So these red ones, that means when arginine was replaced with phenylalanine at 23rd position, then this increased the relative cage formation, and this also leads to the thermal stability. Although if you change it with the tryptophan, then thermal stability increases, but the cage formation decreases. So this also is to show you that with de novo protein design, or with the rational protein design, by bringing about site specific mutagenesis, you can bring about the change in the functions. And this cage study is also one of the examples. So this protein is ECBFR, which is the E. coli bacterioferritin. So this is how the mutation can bring about change in the thermostability as well as cage formation properties of this particular protein. And then there is an Institute for Protein Design which has solved 5 different issues, which have 5 different goals. So they have designed better vaccines using the sequence and structure based design. So they present optimized epitopes on nanoparticles to boost the immune response. This institute is responsible to design the better proteins, or redesign the proteins with functional properties. So they have improved the vaccines by using this sequence based design, conformational stabilization, and structure based designs. Then they also have used the unnatural amino acids to improve the activity of different enzymes. They have also designed the targeted drug delivery methods. So they engineer different protein containers within the targeting domains, in which you can load your drug of your choice, and then this will enhance how the drug get delivered into your body. And these compartments or these containers, they could either be liposomes, or they could be metabolic NPs based on drug delivery. They could be antibody drug conjugates, or they could be polymer based delivery systems. So this is how they have used the protein designing methods to improve the drug delivery technique. Then they have also designed the smart protein switches, in which they can design that whether how the protein will act, so whether it will be in its off state, on state, depending on the signals which are available at that particular point of time. The trigger RNA, it was converting the protein from its off state to the on state. So this is Cas9 protein, which is generally inactive, but as soon as the trigger RNA is present, it converts to on state. So this is known as protein switch. These are also designed. Then nanomaterials for energy. So protein based nanomaterials could be designed, and they could be used in the solar energy or nanofabrication. So these are different applications of protein based nanomaterials. They can help in drug delivery, tissue engineering, wound healing, or purification. So these are some of the applications. And if we talk about the advancements, then ultra potent vaccines have been developed. Cancer immunotherapy has been observed, in which previously the T cell was interacting with tumor, and in this case the T cell was not able to generate or bring about the immunogenic response, but with the help of the drugs or immunotherapies, now T cell is getting blocked, T cell is not interacting with tumor, so now this T cell can bring about the immunogenic response. So all these were helped by the protein engineering techniques. And then rapid antivirals also, they are also designed with the help of protein engineering. So with the help of these antiviral drugs, now previously virus used to enter into our body and infect our body. So with the help of certain inhibitors which can inhibit the fusion of the virus with the cell membrane, there are certain blockers which block these virus receptors. With the receptors it binds to our cell surface. So there are certain blockers which block this entry. So there are certain antivirals which have been designed for H1N1 pandemic strain. Then for the COVID also different antivirals were developed. So this is also the application of protein engineering. Then immune silencing. So by bringing about certain mutations, we can also silence that how the T cell, we can remove the T cell epitope, so which can change the functionality. And then cancer immunotoxin enhancement can also be done, which results in the immunotoxins that retained activity, stability, and anti-tumor effects. So these are also the applications of protein engineering. Then lastly, the tail of heme enzyme. So heme proteins, they are the self-assembling DNA encoded catalysts with tunable electronic and steric properties through evolution. So those proteins contain iron complexes that can be reprogrammed by DNA mutations to perform wide range of chemical transformations. And these engineered heme enzymes, they can catalyze both the natural as well as non-natural reactions. So this include the reactions with abiotic substrates opening up new synthetic pathways. So these heme enzymes, they can be engineered. So for example, this is the biological reagent and then natural selection is done. So there are certain reactions which are known natural. So by using the protein engineering, those known natural reactions can also be carried out. Yeah, so how those reactions can be catalyzed? So we can engineer the certain sites. So firstly, the cysteine was linked to this iron core, but if we replace this cysteine with the serine, then it can also show some known natural reactions. So this is how protein engineering can help you to show certain non-natural reactions as well. This can change certain enzymes which can activate now formation of such bonds which are not possible naturally. And then if you talk about the future direction, then there is supervised learning, and this is a paper by Cadet et al. In which they have shown that how the supervised learning can help you to improve the enzyme activity. So in the supervised learning method, they have performed the point mutations and they have predicted 2n combinations of such single point mutations, and they have shown that how it is influencing the enzyme activity. So if you are interested, you can go through the strategy. So this is the key takeaway messages from this paper, in which they have given a strategy which is INNOV-SAR, which enables the accurate protein optimization using the minimal experimental data and sequence only input. So this method, it overcomes the epistasis challenges by predicting the effect of mutation combinations on protein function, because we have seen that there could be multiple mutations which could be linked to one another. So it streamlines the protein engineering by reducing the need for exhaustive mutation and screening cycles. And machine learning and forward Fourier transformation based encoding, it makes this INNOV-SAR a powerful tool for directed evolution, and this was validated on epoxide hydrolase, on which they have verified this method. So they have predicted the activity of multi-mutant variants, in which not just one mutant but there were multiple such mutants which were present. They were based on the training data, and they have validated 28 mutants with good correlation. That means these mutants were giving they were increasing the enzyme activity. So this is about what are the current approaches which are going on, and this is how they have shown the method, that how they have done it. So they have amino acid sequence, and this is the numerical encoding of this sequence. They have performed Fourier transformation for the protein spectrum. They have multiple such variants, and they are now seeing the protein spectra, the frequency versus the amplitude. So they are comparing the protein activity with the mutants, and then they are selecting those variants which will increase the activity. So with this, they have identified total of 28 such mutants which are giving increased activity. So this is all about how the protein engineering can help you to design those enzymes which are having the better catalytic activities. So now we'll move to practice MCQ questions. Yeah, so now first question is, what does the Ramachandran plot show? And for example, first option is supersecondary structure, option B is frequency of hydrogen bonds in proteins, option C is domains in proteins, and option D is frequency of phi and psi values of residues. Which option? D? Okay. Yeah, so D option is right, because it shows you phi and psi distributions, and then it helps you to see that which kind of secondary structure the protein sequence can adopt. Yeah, and this is a detailed representation. So if you're interested, you can go through this plot. Next question is, in Rosetta, the segments with assigned secondary structures are subsequently assembled into which dimensional configuration? Option A is 2, option B is 3, option C is 4, and option D is 6. Which option? B or D? So the right answer is option B, that in Rosetta you assemble the secondary structure segments into 3D protein structure. Now next question is, which of the following is responsible for specifying the 3D shape of a protein? Option A is interaction with molecular chaperones, option B is peptide bonds, option C is interaction with other polypeptides, and option D is amino acid sequence. So you have to tell which one is. So the right answer is option D, that is the amino acid sequences. So we have already discussed that the 3D shape of a protein is determined by the amino acid sequence. So primary sequence governs that what will be the 3D structure, because in protein engineering what we are doing is we are changing the amino acid sequence and then we are bringing about change in the protein function. So if we talk about other options, then molecular chaperones, they are generally involved to improve the protein fold. If protein is misfolded, then chaperone helps to prevent that misfolding and it folds them properly into three-dimensional structures. Peptide bonds, these are simply the bonds which are linking two amino acids together. So they are not responsible for specifying 3D shape because they are anyway present between the amino acids. And then interaction with other polypeptides, so they contribute to the multi-subunit complexes but not to the 3D structure of a single protein. So next question is, which factors contribute to the stability of protein structure? Option A, hydrophobicity. Option B, electrostatic interactions. Option C, disulfide bridges. And option D is all of the above. Okay, yeah, right. So D is the right, because we have seen that these are different interactions which are generally observed, which are responsible for the stability. Next question is, which of the following is a component of PCR reaction? Option A, DNA ligase. Option B, DNTPs. Option C, RNAs. And option D is DNAs. So you have to tell which one is a component. Okay, let's see the answer. So the right answer is option B, which is DNTPs. So in PCR we have seen this is a polymerase chain reaction in which these are different components which are required. So DNA ligase is not generally used in PCR. RNAs are the enzymes which degrade the RNA, so they have nothing to do with the PCR. And DNAs are also the enzymes which degrade the DNA. So in PCR we are amplifying the DNA, so we do not need these enzymes. We need DNTPs along with all these components. So now next question is, the rational design of new protein molecules to design novel activity, behavior or purpose and to advance the basic understanding of protein function is called as. Option A, protein design. Option B, protein instability. Option C, protein unfolding. And option D is protein cleavage. Okay, we are seeing option A. Yeah, right. So we've already seen that the rational design comes under protein design, in which we use the computer aided information. So we already have all the structural information of the protein and simply we perform certain site directed mutagenesis and we change its functions and properties. So next question is, which of the following is not true about the ab initio predictions? Option A, the limited knowledge of protein folding forms the basis of ab initio prediction. Option B, the perceived advantage of this method is that predictions are not restricted by known folds and that novel proteins fold can be identified. Option C, the ab initio prediction method attempts to produce all atom protein models based on sequence information alone with some aid of known protein structures. And option D is none of the above. So you have to tell which is not true. Okay, let's see. So right answer is option C. A is right. The limited knowledge of protein folding forms the basis, because we do not have the entire knowledge of all the protein folded structures. So for that reason we need to go for ab initio prediction. So this is the limited knowledge is the basis why we have moved to ab initio predictions. And in the second also they have mentioned that the perceived advantage of this method is the prediction is not restricted by known folds. That is the main basis of ab initio, that we are not limited by the information. We can, in ab initio, we predict from ourselves by using the different laws of physics. But the C option, which was that the ab initio prediction method attempts to produce all atom protein models based on sequence information alone with some aid of known protein structures. So this one is not true, because ab initio by definition it typically avoids using the non-structural templates. So including the aid from the non-structures aligns more with the hybrid or template based methods. So it does not use any known protein structures. Next question is, what is the de novo design in protein engineering? Option A, designing of protein with few mutated residues in the original sequence. Option B, designing a new sequence based on prior sequence, option C both of the above, and option D is none of the above. Okay, let's see. So the right answer is option D, and in the previous assignment they have said that option B is also right. So we'll discuss that why. So in first option they mentioned the designing of protein with few mutated residues in the original sequence. So de novo means when we start from scratch, we do not have any template. So option A is completely irrelevant for the de novo protein design, because we are not working with the original sequence. We are designing entirely new sequence. But when we talk about option B, then designing a new sequence based on prior sequence. So it depends how we interpret it. So if the prior sequence means a template, then this option is not true. But if prior sequence means that we are taking information of already available secondary structures or already available protein domains or protein parts, then this option can be true. So it depends on how you interpret it. So in that way, if option B could be right, if we only consider that this prior sequence means of these small stretches, then this is true. Otherwise, de novo means creating entirely new protein sequence from scratch, often using computational methods without relying on any existing templates. Now next question is, ITESR structure prediction is based on option A homology modeling, option B super secondary structures, option C multiple threading approaches, and option D is phylogenetic resemblance. Yeah, so C is the right option, multiple threading approaches, in which we have seen the Lomet's threading which it uses for the entire sequence. It see which of the regions which have already designated folds. So firstly it perform multiple threading, and then it combines ab initio methods also to predict the entire structure. So yeah, this option is right. Now the last question is, change of one nucleotide, that is A to C, in the gene sequence of an enzyme is known as option A deletion, option B substitution, option C large modifications, and option D is insertion. Yeah, so you're right. The right answer is option B. So this type of change is known as substitution, when one nucleotide is replaced by the another nucleotide. This is substitution. If there is some insertion, then that is insertion. If some nucleotides are lost, then it comes under deletion. Yeah, so this is it for today's session. So if you have any doubts, then you can ask, or whether I'll end the class. Okay, so if you don't have any doubts, then I'll end the session, and I'll see you in the next session for the week 12. So thank you very much.